Hello everyone, welcome to today's presentation. My name is Savannah Hinchin and I'm with EOS Remediation. Today's topic is applications of BioTraps for environmental site diagnostics. And our speaker today is Dr. Carey Sublett with the University of Tulsa's Chemical Engineering Department. He co-founded the BioTrap technology along with Microbial Insights. So we are very fortunate to have him speaking today. Now, before we get started, I'd like to share a little bit about EOS Remediation and what we do. EOS is the original developer of the emulsified oil technology for in situ bioremediation. We are located in Raleigh, North Carolina, and have been in business since 2002. A few announcements. Today is our first webinar in the 2014 and 15 webinar series, so make sure to pay close attention to your email. We'll have webinars every other month up until, I believe, May of next year, and then start over again. I want to remind everyone of our EOS design tool. It's available through our website, www.eosremediation.com. You will have to be logged in to use it, and don't forget to keep going through the pages until you can submit your final design, and Brad and I and the rest of the EOS Remediation sales team can help you with the information as well. So we do have a couple international events coming up. We are attending Big Site in Tokyo. That's October 15th through 17th. Ed Alfred will be out there. And we are also conducting remediation and MBT workshops with Microbial Insights in Sydney and Melbourne, Australia on October 20th and the 27th. And keep a lookout for us at the next domestic conference in Orlando, Florida. It's FRC on October 9th and 10th, and I believe Brad will be there. So look out for him. Now I'm going to pass the torch over to Carey and get this thing started.
Okay, thank you, Savannah. As Savannah indicated, my topic today is application of biotraps, particularly for environmental site diagnostics. And probably the first step to take here is, if I can get, there we go, find a way to advance my slides here, is to tell you what a biotrap is. For those of you that may not be familiar with biotraps, are a passive sampling tool. And by a passive sampling tool is for microorganisms. And by passive, what I mean is that you install a biotrap in an environment to be sampled, and you basically leave it there to allow biofilms to be established inside the medium of the biotrap, which is the biosep beads that you see in the picture here, that I'll tell you more about in just a few moments. But you leave that in place typically 30 to 45 days, and then you retrieve the biotrap for analysis. So basically there's no care and feeding during the incubation time. It's important to understand what it is that a biotrap will collect. As I said, that during the incubation period, biofilms are formed on the inside of these beads. Biotraps collect only the active component of the microbial population in the environment that's being sampled. And typically that environment is going to be a groundwater sampling well, or a groundwater monitoring well rather, but it can be any of the many different types of environments, as you'll see. But basically, before an organism can actually get into the bead and participate in the formation of a biofilm, it has to be actively growing and reproducing in that environment during the time of sampling. So what you're going to be seeing then when you analyze the biofilm inside of a biotrap and inside these biosep beads that I'll be telling you about are those organisms that are, excuse me, something just popped up. There we go. What you're going to be collecting are the active component of that microbial community that is being sampled. So as I was saying, to get into the beads, to participate in the formation of a biofilm, and therefore to be collected and detected and analyzed, these microorganisms have to be growing and reproducing. So we know in any sampled environment, groundwater included, there are going to be a lot of microorganisms around that aren't necessarily growing and reproducing at any given time. If they're not, because they're waiting for the right nutrient to come along or the right environmental condition to come along, if they're not growing and reproducing, then basically they're invisible to the biotrap. Now that's a big advantage, because when it comes to investigating or looking at the microbial community structure in an environment like a contaminated aquifer, what we're really interested in are the microorganisms that are involved in the transformations that are going on in that aquifer, and those are going to be the organisms that are active in growing and reproducing in that environment. Another thing to remember about a biotrap is that it provides an integrated sample versus a snapshot. If I go out to a groundwater monitoring well and take a sample of that groundwater, and I take it to the laboratory and analyze it, that is a snapshot of a point in time. Not only will I be looking at a mixture of the organisms that are actually involved in the transformations that are going on and growing and reproducing, I'll be looking at all those organisms that are not as well. But I'll also be looking at a point in time. Well, the biotrap being incubated typically in a groundwater monitoring well for 30 to 45 days, basically this biofilm that's forming on the inside of the trap is coming into equilibrium with that community over that time period, and therefore we get an integrated picture of what's going on in that sampled environment. Now, once the biotraps are retrieved, we can analyze these biotraps using a variety of different molecular biological tools, also analytical chemistry, stable isotope analysis, as you'll be seeing. Now, how do biotraps work? The heart of the biotrap is the BioSep bead. It is a proprietary medium. It's proprietary to Microbial Insights in the University of Tulsa. Basically, the BioSep bead is about 3 to 4 millimeters in diameter. You're seeing a cross-section, low magnification picture of a cross-section in the upper right-hand corner here. They're made out of two materials, Nomex and powdered activated carbon. Nomex, you might recognize, is the primary polymer that's used in first responder suits, so it's very thermally stable, and it's made by DuPont. So basically the Nomex provides a framework, or a network we might say, for holding particles of powdered activated carbon, and it's on these particles of powdered activated carbon where biofilms are formed. The porosity of the beads is very high. There's a lot of surface area there available for colonization. And one of the really nice features of these beads, because of the Nomex, is they're very stable to high temperatures. And the benefit of that is that we can heat these beads to 270 degrees centigrade before they go into a biotrap. And in doing so, we not only sterilize the beads, but we also destroy fossil biomarkers, and we also drive off any organics that might have, from the atmosphere, been absorbed to the powdered activated carbon surface. So you can be sure that when you get these biotraps, the beads inside are clean and sterile. The other pictures that you're seeing on this slide, the picture in the middle here is a higher magnification picture of a biofilm on the inside of a BioSep bead. And this is just a similar picture on the bottom here, but this is a live dead stain. Now, there are many different types of biotraps, but I'll first of all introduce you to the most common type. This is a standard biotrap. Basically, this is a piece of slotted half-inch Schedule 40 PVC with these PVC end pieces, and is packed with BioSep beads, typically on the order of 200 or so in one of these tubes. So as I said, this is half-inch diameter. It's about 4 inches long. This is the standard design. If you were to call Microbial Insights and request these biotraps for just standard monitoring purposes, say in a groundwater monitoring well, this is what you're going to receive. Now, there's other types of biotrap designs. For example, Microbial Insights also markets what they refer to as an in situ microcosm. which is an enclosure like the one that you see here that contains several different things, including a biotrap. This particular biotrap is about 3 inches long and about 1 inch in diameter. But in these in situ microcosms, you can also have other types of passive sampling devices for contaminants, for geochemistry. You can also provide remediation amendments of one type or another, like EOS's products, for example, in these in situ microcosms. They then get deployed in a groundwater monitoring well, typically in either pairs or in triplicate. In the example that I've shown here, we've got one here I've marked control, one treatment option 1, treatment option 2. The control might contain a biotrap and passive diffusive samplers for the geochemistry and the contaminants of concern, whereas treatment options 1 and 2 might also contain a supply module to supply a potential remediation amendment that you're interested in looking at. And I'll be giving you some examples of deployments of these types of in situ microcosms and their inherent biotraps a bit later. Now, these in situ microcosms are baffled at the end pieces so that when they're deployed, they're isolated from one another in the well to minimize any crosstalk that might take place. The various types of amendments that have been used in these in situ microcosms, along with the biotrap, various types of vegetable oils, molasses, EOS products, lactate, HRC, different kinds of electron acceptors, and as you'll learn here a bit later, many different kinds of stable isotope compounds, from hydrocarbons to fuel oxygenates to chlorinated hydrocarbons, and some emerging contaminants like 1,4-dioxane and sulfolane. Now, a biotrap is typically going to be deployed in a groundwater monitoring well, but as I said earlier, that's not the only environment where these are deployed, but it's just the most common. And typically what you're going to do is purge the well as you might do before sampling the groundwater, and then you would suspend the biotrap from the top of the casing, and you would simply suspend it typically in the center of the screened interval, or if there is a particular depth of interest, you could suspend it at that depth. If you are expecting large fluctuations in the water level in the monitoring well, you can also suspend the biotraps from a float. So as the water level goes up and down, that the biotrap is maintained at a given distance from the interface. In terms of where we deploy biotraps, if we've just got a hypothetical plume here, probably the ideal deployment would be along the axis of that plume, including, if possible, a background sample upstream of the plume. Or if you've got sufficient budget, also deploying downstream or down gradient, as well as on the fringes of the plume would be advisable. There are different sampling strategies over the life of a plume. If you're monitoring, for example, a remediation scenario that's going on, you might deploy a biotrap in a given well, retrieve that 30 days later, replace it with another biotrap, retrieve that, replace it, etc. Or there are also ways that you can deploy multiple biotraps in a well and then retrieve those biotraps at whatever sampling interval that you're interested in. Now, those are the standard designs for biotraps, the most common designs, but there are a lot of specialized designs too that have been used in different types of environments. For example, here are some flow-through biotraps that were used on a slipstream in a drinking water distribution system. Here you're looking at biotraps that were deployed in a solvent extraction system and basically a drum sampler so that they could be, you could go down through the top kerosene layer and then open up the sampler and expose the biotraps to the aqueous layer underneath. These are some biotraps that you see here are mounted on a corrosion coupon holder. These were deployed in a pipeline in Alaska. Here's another type of biotrap in which we can actually, during incubation, we can feed the biotrap with a compressed gas. Usually that's going to be something like a source of hydrogen, or it could be just aeration. And so basically what you have here is a sparger in the center of the trap, and it's surrounded by biosep beads. And here's, for example, a couple of these types of biotraps that were deployed, each one of these connected to a different oxygen concentration in the compressed gas on the surface, so that we could create different dissolved oxygen levels in the biotraps. Here's another one that has been used in streams as well as on the ocean floor. It's basically a sediment sampling device. So basically the biotrap, as you can see here, this is just kind of like a standard extra long biotrap, suspended from the cap of this samplers at the next to the perforations here, and then the entire thing can be hammered down into the sediments again on a stream, or in one case these were hammered into the sediments in the ocean floor by divers. Here you're looking at some standard as well as some oil-emitted biotraps that were deployed in the Gulf of Mexico after the BP oil spill. These were being deployed at a depth of about 5,000 feet and on one of the risers there, and were used to help study the microbial populations and the response to the release of hydrocarbon into the Gulf. No matter what kind of biotrap you've got, once it's retrieved, you've got a lot of options as far as the analysis of the biofilms, phospholipid fatty acid analysis, qPCR, the new quant arrays, which you may or may not be familiar with, but basically it's a quantitative multiple qPCR tool. Older tools like DGE, stable isotope analysis for stable isotope probing, which I'll be telling you about shortly. But also, too, these beads can be extracted for different types of chemical analysis. Common things that are done is, for example, with chlorinated hydrocarbons. Frequently, chlorinated hydrocarbons are extracted out of the beads after incubation for stable isotope analysis. Another common analysis is to extract dissolved inorganic carbon from the beads, also for stable isotope analysis. So I'd like to address now different things we might do with a biotrap sampler, and I've got basically 4 bullet points here. We'll talk about the first one, determine if known degraders of a contaminant of concern are present at a site. And just to give you a quick little example, here are the results of analysis of biotraps that were deployed in a site impacted by chlorinated hydrocarbons, 2 different wells. So here, of course, with a chlorinated hydrocarbon, we're interested in the concentrations of dihalococoides and also reductive dehalogenation activity. So what you're looking at here at these 2 sites was an analysis of the biotraps for dihalococoides, as well as, in this case, vinyl chloride reductase genes. And as you can see in this particular case, both dihalococoides and vinyl chloride reductase were identified at this particular site. Now, if I go on to the next 2 bullets, I'm going to basically combine those. But we can evaluate, monitor natural attenuation versus enhanced bioremediation. We can also compare the effectiveness of amendments designed to stimulate biodegradation. And in doing so, we're going to use those in situ microcosms with the 1-inch biotraps I was telling you about. And these are used in basically 2 different types of sites, typically either a hydrocarbon impacted site or a chlorinated hydrocarbon impacted site. To give you some examples of how these might be used, for example, at a petroleum contaminated site, we might deploy a control microcosm, an oxygen amended microcosm using an oxygen release compound, and we might compare that, for example, to a situation in which sulfate may be added as a terminal electron acceptor. So if I deploy these and then incubate them for a suitable period and then recover the biotraps for analysis, what the control biotrap will tell me is what the microbial community looks like now without either one of these amendments. The oxygen amended microcosm, the biotrap from that microcosm will tell me how is that microbial community going to change now if oxygen is made available. And of course the same question down here with the sulfate, how is that microbial community going to change if sulfate is made available. Over here on the chlorinated hydrocarbon side, again, what I've indicated here is again another control without any kind of remediation amendment. The second one is indicated here might be an enhanced anaerobic remediation amendment, say like an EOS veg oil, emulsified veg oil product, for example, that I would call biostimulation. And we could compare that to another product, or what I've indicated here is a bioaugmentation unit. We know that there are several commercial cultures that are available for dihelicocoides, and we've been asked at times to evaluate the fate of those cultures when they're put into a particular aquifer environment. So the biotrap that's in this particular unit can be pre-incubated with that commercial culture to establish a population of those organisms in the biotrap, and they can then be incubated in the aquifer, and we can document the effect that they might have on reductive dehalogenation, but we could also determine the survival of those organisms when they're being incubated in the actual in situ environment. So to give you an example of this use, here we're looking at a site impacted by chlorinated hydrocarbons, and we have a control and an amended unit here with some sort of electron donor. And comparing the control to the amended, you see that making this particular amendment available increases the population of dihelicocoides, but it also increases the number of vinyl chloride reductase genes that are being detected as well. So these types of results would indicate then that this remediation amendment should have a positive effect both on the growth of dihelicocoides and at the same time reductive dehalogenation if it is made available to that in situ population. To further illustrate this, I'd like to give you a case study now. This particular case study is a site impacted by chlorinated solvents in upstate New York. To give you a little background, the contaminants of concern here are primarily going to be PCE, TCE, and cis-DCE. There's also some TCA and methylene chloride as well. The site had been treated by pump and treat for about 4 years with over 9,000 pounds of chlorinated hydrocarbons removed. So the client here was interested in perhaps then transitioning to either monitored natural attenuation or an enhanced attenuation option. And in this particular case, there is no immediate risk identified, no plans to sell. The state was stable plume, minimal vinyl chloride and ethene, though, had been detected. There was a buildup of cis-DCE. So basically, they wanted to evaluate the potential for enhancing remediation using an electron donor. But we've got some problems here, though. These are some analyses of 2 different groundwater samples from 2 different wells in this particular site. And what I wanted to point out to you is the fact that the sulfate concentration at this site was quite high. And we know that to deploy an electron donor at a plume impacted by chlorinated hydrocarbons where the sulfate concentration is elevated, we run the risk of stimulating sulfate-reducing bacteria, which will not only produce hydrogen sulfide, and which will be potentially inhibitory to the organisms in the aquifer, but also the SRB then basically are going to be, as they're stimulated by the electron donor, they're going to be competing for that electron donor with the organisms we'd like to stimulate, which is the helicocoides. So because of this, at this particular site, the client wished to evaluate a couple of different remediation amendments. One was just standard HRC, but the other was a product that was under development that I understand is a commercial product now, though not under this name, that they refer to as HRCS. And the intent of the HRCS was it was designed specifically to stimulate reductive dehalogenation in sites with high sulfate concentrations. The idea here was that this particular formulation also contained a chelated iron 2, and the purpose of which was to bind and precipitate sulfides that are produced by sulfate reduction, and in doing so basically take that sulfur out of the sulfur cycle. Otherwise it could be easily reoxidized as sulfate by thiobacilli, but by precipitating with iron, the intent was to take it out of the cycle and take that sulfur away from the sulfur cycle, and then eventually remove the competition with sulfate-reducing bacteria. At least that was the strategy, that was the idea. So here's a site map of the site, and several wells 4 different wells here were chosen for study with different levels of CVOC concentrations from 100 part per million down to 1 part per million, and then another well back here which was up gradient where the CVOCs were not detected. So what happened here was we deployed BioTraps or in situ microcosms that one of which was a control with no remediation amendment, another with an HRC amendment, another with an HRCS amendment. And you're looking first of all at the results as far as dihelicocoides concentrations are concerned. And what you see at the 4 wells that are being sampled here, there was some stimulation of dihelicocoides with just HRC in 1 well, but not in the others. But with HRCS, dihelicocoides was stimulated in all of the other wells, including the background, at concentrations quite a bit higher than was produced by HRC alone, even in that 1 well. So that right there indicated that, that the HRCS did seem to be a better source of electron donor to stimulate dihelicocoides in this particular environment. But we went further and looked at concentrations of sulfate-reducing bacteria, and what I want to show you here is that, first of all, this is a log scale, so we've got significantly lower concentrations of sulfate-reducing bacteria in the presence of HRCS compared to HRC. Again, once again, indicating that the intended effect of preventing the stimulation of sulfate-reducing bacteria or limiting that stimulation does seem to be a characteristic of this particular electron donor. We further looked at concentrations of methanogens and found that no matter what is the electron donor, we do get a stimulation of methanogens with either HRC or HRCS, kind of providing a red flag here indicating that perhaps you're going to see some competition from methanogens with either one of these remediation amendments. So to summarize, HRCS was identified as the most effective at stimulating dihelicocoides. We did get some help in controlling SRBs with the HRCS, but methanogens can still be a problem. The entire study took about 75 days. That was 60 days of incubation, about 14 days of analysis. The cost was 1/3 of that of the traditional microcosm study. Now the next step in this process was to do an HRC injection pilot, and I'd like to show you some of that data this is the biostimulation point right here. And interestingly enough, what you see is the DCE concentration goes up, the vinyl chloride concentration stays about the same over the first 100 days. But then we see the DCE concentration going down, but the vinyl chloride concentration is not going up. If we superimpose on that the microbial results, we see that accompanying that increase in the DCE concentration was actually a decrease in the concentration of the dihelicocoides. But at the same time, there was an increase in the concentration of methanogens. So clearly what seems to be happening here, that in the first 100 days or so, the methanogens seem to be outcompeting dihelicocoides for the available resources. If we go on in time, now we see the DCE concentration starting to go down and the vinyl chloride concentration starting to go up. We're also starting to detect ethene. And once again, if we look at the microbial results, we've seen the accompanying this favorable result in the COC concentrations. We're seeing the concentrations of methanogens going down, and now we're seeing the concentration of dihelicoides going up. So initially, while we were seeing competition from the methanogens that was limiting the growth of dihelicoides, as time goes on, we see dihelicoides now starting to outcompete the methanogens. And as time goes on, now we see the vinyl chloride concentration going down, ethene concentration going up further. And then if I simply summarize the results from this pilot study, as you can see, overall after the addition of or the injection of the HRCS, there is a large and now sustained stimulation in the dihelicoides population, as well as as seen here, vinyl chloride reductase. So they proceeded on with a full-scale injection of HRCS at this point. Another case study now with baited biotraps or in situ microcosms, now a petroleum hydrocarbon site. We're looking at a shallow aquifer impacted by gasoline here, low dissolved oxygen concentrations, low concentrations of alternative electron acceptors. So the site-specific question the client wanted to answer here was, will oxygen addition promote the growth of bacteria capable of aerobic biodegradation of hydrocarbon at the site? So what was done at the site was to deploy in situ microcosms, controls versus an oxygen-amended microcosm. And just to show you some of the results at some of the wells at this site, here we're looking at, these are concentrations of phenol hydroxylase. Again, all three of these are enzymes involved in aerobic biodegradation of hydrocarbons. And what you see is you compare this enzyme, or this gene in the control versus that in the oxygen amended. And I'll remind you again, you're looking at a log scale here. As you can see, a couple of orders of magnitude increase in the concentration of that gene in the presence of oxygen. Similar results with the toluene dioxygenase, and similar results with the xylene monooxygenase as well. In fact, it was not detected in the control unit. It was detected in the oxygen-amended unit. Another case study. This is an aquifer again impacted by petroleum hydrocarbons. In this case, standard biotraps were installed in this aquifer, and we were using DGGE to try to predict what effects sulfate injection would have in the aquifer. So we had biotraps that were amended with sulfate, and those that were not amended with sulfate. Here we are in one of these plumes, and what you're looking at here is just an XY scan showing the intensity of the different bands in the DGGE gels. And here in this particular monitoring well without sulfate, we see one organism that's identified here that's a potential sulfate reducer, Geobacter. But in that same well, over the same incubation time, in the sulfate-amended biotrap, we see a number of different Geobacter species, and many of these species are known to be able to use sulfate as a terminal electron acceptor. So following results like this, there was a sulfate injection gallery installed, and then we monitored what was going on in the aquifer, again using biotraps. And what we see over time here, you're looking at SRB populations going up, but also we're seeing concentrations of the BSSA gene, which is, if you're not familiar with this, this is a gene indicative of anaerobic degradation of substituted benzenes. You see it popping up as well. So getting down to the last bullet point then, proving that bioremediation of a specific compound is occurring under in situ conditions. Basically, what I'm referring to here is stable isotope probing. Stable isotope probing is a technique using biotraps that we can do two things with. Number one, we can provide basically irrefutable proof that biodegradation potential of a given compound exists under in situ conditions. And then with further analysis, if desired, we can also link that biodegradation potential to specific microorganisms that are involved in the process, and I'll illustrate that for you. So basically what we do is, in stable isotope probing, we use C13 labeled compounds. Now, you're probably familiar with C13, but it is a heavy, it's not a radioactive isotope. It's a slightly heavier isotope of carbon that just has one additional neutron in the nucleus. So it's just heavier than normal carbon. Typically, 99% of the carbon in the biosphere is going to be carbon-12, and about 1% is going to be carbon-13. What we do is to artificially increase the concentration of carbon-13 in a particular contaminant of concern. And then we basically, using a biotrap, offer that up to microorganisms in an environment to see if anybody there is capable of biodegrading that compound. Now, before I go further and show you some results, we need to distinguish stable isotope probing from compound-specific isotope analysis. These are very different techniques. With compound-specific isotope analysis, you're looking at isotopic fractionation of naturally occurring C12, C13 mixtures in the environment. So you're looking for enrichment in compounds that are undergoing biodegradation. You should see an enrichment in the amount of C13. Stable isotope probing is very different because now we're intentionally introducing carbon-13 as a tracer, and we're following where that carbon-13 is going to go. We look for it in CO2, indicating mineralization. We can look for it in methane under anaerobic conditions, and we look for its incorporation into biomass. The most popular thing to look at, and the easiest thing to look at, is phospholipid fatty acids. But if we offer up this carbon-13 to a microbial population and we see incorporation of that carbon-13 into CO2, methane, and into biomass, there's only one possible explanation, and that is that the organisms present in that environment can and did biodegrade that compound and mineralize it and utilize it to make more microorganisms. So basically the way this works then is we take a bioset bead and we adsorb a C13 compound onto that bead. Here we're looking at benzene. And then we incubate that in an environment where there's microorganisms that have the potential for benzene biodegradation. And if they do, then they will metabolize that benzene and incorporate benzene into their biomass, and they will also produce products of mineralization of that compound like CO2 or CO2 and methane if we're under anaerobic conditions. The beads are loaded under vacuum, and basically the C13 compounds are adsorbed onto the powdered activated carbon component of the beads. Here's another representation of the technique. The C13 labeled compound is adsorbed to the beads. That goes into a biotrap. That goes into the sampled environment. After incubation, we recover the beads for analysis. One question you might be asking yourself is, if we put something like benzene on the beads, what happens to that benzene? Does it stay in the beads or does it leach off the beads? Here's an example of some data showing you this is just normalized concentration of benzene on the bioset beads for 30 days of incubation. And as you can see, none or very little of that benzene actually leaches off the beads. It's held by that powdered activated carbon in the bead. So once we recover the biotrap, we do 3 different types of analyses. As I alluded to a moment ago, we look for C13 incorporation into CO2 and methane. We look for C13 incorporation into biomass. And we can also determine how much of the C13 labeled compound is still on the beads, and that gives us kind of an idea of relative rates. So I'm running kind of short of time here, but basically one of the things that we can do with this that helps us get some information about what kinds of organisms are involved in the biodegradation event. The incorporation of C13 into the lipids, there are various types of lipids about 16 different common lipids, for example, that are found in cell membranes. And there's information I'll point out to you that's on MI's website, different structural classes of phospholipids and what kinds of organisms are characteristic of these different structural classes. So if we see which one of these classes are incorporating carbon-13, it can also give us some information about what organisms are actually doing the biodegradation. And I guess I'd like to point out at this point too that we can also, it's a little bit more difficult to do but certainly doable, we can look for C13 incorporation into DNA as well and see what genes that C13 has been incorporated in, and from that we can then identify what types of organisms are again involved in the degradation process. You may be familiar with the unit of measure for C13 composition of an organic compound, but basically it's a del 13C notation that is computed by looking at the C13-C12 ratio of a sample. Compare that to a standard. The standard is PD belemnite. It's a mineral. And then we calculate the del 13C value from this equation. And basically what that results in then is a scale, a del 13C scale, on which the standard has a value of 0. And then anything with a value less than 0 contains less carbon-13 than the standard. Anything with a value greater than 0 contains more carbon-13 than the standard. And on this scale here, I've just shown you some common materials like oxygenase, chlorinated hydrocarbons, benzene, crude oils, natural gas, etc., and the del 13C values for these materials as they naturally exist in the environment. When we use C13 as a tracer and we incorporate carbon-13 into either products of mineralization or biomass, we're going to get results that, as you'll see, are far to the right. So there will be no misinterpretation of what we're looking at. We'll be able to see carbon-13 incorporation from the tracer very clearly, as you'll see. So I'll end up with this case study. Here we're looking at paraxylene at a site in New Jersey. And basically the client was interested in two questions. Is there paraxylene degradation that's going on now under the current conditions? And to what extent could I stimulate paraxylene biodegradation with injection of oxygen? So what we did here was to install BioTraps samplers that were amended with C13 paraxylene at 3 different wells with 3 different background paraxylene concentrations. They were deployed for 30 days and then retrieved and analyzed. This is a map of the site showing you, first of all, 3 wells where BioTraps were deployed that were under the influence of an air sparging system. And then there was another well back here where BioTraps were deployed where there was no oxygen communication to that well. So that's our background or our control. Just to show you some results from the site, after retrieving the BioTraps, we knew exactly how much paraxylene was on the beads when they went out the door, and so that we can also measure the paraxylene concentration on the beads as they returned, and we can see the amount of loss of paraxylene during incubation. And in the control, we've got a 6% loss, but as you can see, 16 to 40% loss in the presence of oxygen. So that's the immediate evidence we have that oxygen is stimulating biodegradation. Here now we're looking at the del 13C values for dissolved inorganic carbon extracted from the beads. Here are the 3 wells that were biostimulated with oxygen. Here's the background control. As you can see, we've got a pretty large positive value for the del 13C value. Even in the background, the control, or the M&A control, we might say. So there's definitely paraxylene biodegradation that's going on without an oxygen amendment. But if you add an oxygen amendment, you see how much higher the DEL values have gotten, indicating much greater incorporation of carbon-13 into CO2. So definitely the SIP results are indicating that oxygen will stimulate the mineralization of paraxylene. If we look further then at the biomass, in particular looking at the phospholipid fatty acids for the control and for the biostimulated, these are average, minimum, and maximum results in the different fatty acids that we recover from the beads. And as you can see, there is definitely some incorporation of carbon-13 in the absence of oxygen, but in the presence of oxygen, we've got tremendous stimulation of the degradation of paraxylene and the incorporation of carbon-13 into the biomass. Now, I'd like to show you, I'm just going to skip to this since I'm short of time here, but I'd like to show you a little different way of looking at these results. Here I'm looking at the common fatty acids that I find within the phospholipids and typical bacterial cell membrane. And the line that I show here is at 1.1%, which is approximately the background concentration of carbon-13. So now I'm looking at the atomic percent of carbon-13 in each one of these fatty acids. So if a fatty acid has incorporated carbon-13, it will have a atomic percent C13 that is greater than 1.1%. So if they peak above the line here, then there's been incorporation of carbon-13. This is the control. So you can see there is some incorporation that's not terribly high. Here is one of the oxygen amended ones here. As you see, the bars have gotten a lot higher. And here's at the other well. And here's at the third well where we're seeing the greatest activity with oxygen amendment. Now, my reason for showing you this is because, again, using the information like that that's on the MI website, we can determine what kinds of microorganisms are actually involved in the biodegradation of paraxylene. And if you look at that, you see that there are anaerobes involved, there are aerobes involved, there's even a sulfate-reducing organism that's involved, and there's fungi involved. So what we're seeing here with oxygen stimulation, a huge community of microorganisms are being stimulated and participating in the biodegradation of paraxylene in that well. In closing here, I would like to point out that MI maintains a database of, in fact, they're going to be going live and online with this database by the end of the year, in which they can provide for you information about other people's experience with different types of C13 labeled compounds like the ones that you see here, benzene, MTBE, TBA, naphthalene, chlorobenzene, methylene chloride, and this list is expanding all the time. Looking at percent removal of the compound from the bead during incubation, highest DEL13 values for the fatty acids, highest DEL values for DIC. The bottom line is that they have a lot of experience at this. They've been doing this for over 10 years. Lots of clients have deployed lots of SIP biotraps with lots of different types of amendments, and then they can communicate to you very readily, how do your results compare to what other people have seen under similar incubation conditions. So with that, I'm going to close and address any questions you might have. Here is my email address. If you would like a copy of this presentation, all you need to do is send me an email, and I'll send you a copy by return email. So with that, I'll be happy to take questions.
We have a message, there are questions that says biotraps for active bacteria. What is used for non-active?
The only, as far as providing an integrated type of picture, I don't know of anything really that's available. Certainly snapshot type pictures of a point in time are readily available just by taking a groundwater sample, taking a soil sample, etc. Now when you analyze that sample, you're seeing everything. You're seeing those organisms that are active and those organisms that are not. I think comparing that type of sample with a biotrap sample, for example, and then by difference, you might be able to then infer what organisms are there that are not active.
Okay. The next one is the results or are the result beads also comparable to cell in the soil and groundwater?
Well, no, this is you were talking about the concentrations. When it comes to Dihalicoides, one of the observations that has been made is that the concentrations on a per bead basis seem to be pretty similar to the concentrations in groundwater on a per milliliter basis. For other types of organisms, that could be different. But the concentrations in the beads are always reflecting what the concentration is in the sampled environment. The higher the concentrations in the sample environment, the higher the concentrations are in the beads and vice versa.
Okay, thank you. Have biotraps been used for bird technology projects where stimulation of sulfate-reducing bacteria is optimized?
I'm reasonably sure that is the case because I know MI has had a number of clients, for example, that have been using or have used sulfate injection to stimulate degradation of chlorinated hydrocarbons, and they have used biotraps to monitor the progress of that stimulation. And in some cases, it's been coupled with stable isotope probing to show, for example, that an actual injection of sulfate as a site has increased the degradation potential of a particular target contaminant. Typically, that's been benzene.
Carrie, I can follow up with that as well. This is Brad with EOS Remediation. We regularly, as long as your scope, schedule, and budget allows for it, if you were to be looking at using something like our EAS technology, which is a sulfate technology, I typically recommend to the project team that they highly consider deploying a biotrap at their site so they can confirm whether or not they have the sulfate-reducing bacteria on site.
Thanks, Brad. The next question we have is, what is the cost of the biotrap and the lab analysis?
Well, it depends on the analysis that's being done, and it's a little dangerous for me to cite costs because I don't get involved in that. But I can tell you that I believe that the biotrap itself, a standard biotrap, is on the order of around $100 or so. Then it depends on what analyses that you want. A phospholipid fatty acid analysis, I believe, is $375. Please don't quote me on these numbers if MI gives you something a little different. As I recall, the cost for quantitative PCR on the biotraps are exactly the same as they would be on any other sample, and I believe that is around $250 for the first target and $50 for each additional target. Or if you do a quant-array qPCR where you might be looking at 100 different targets at once, MI has several different types of quant-arrays for different types of contaminants, from petroleum hydrocarbons to chlorinated hydrocarbons, etc. And I believe those run about $750. So that if you get more, if you're interested in more than 7 or 8 targets, like different taxonomic genes or different functional genes, then it becomes much more cost effective to use a quant array. Now, as far as stable isotope probing is concerned, that depends very much on the compound, because as far as the trap itself is concerned, the primary element in the cost there is the cost of the C13 labeled compound. A lot of hydrocarbons, monoaromatics, for example, are readily available at very reasonable costs. So I believe that the cost of a biotrap with C13 benzene, for example, and all of the analysis that's required of that after incubation would run around $1,500. And then as the compound gets more unique, the cost will go up, but that, again, that's because of the cost of the C13 labeled compound. But most of the common contaminants, from chlorinated hydrocarbons to hydrocarbons to oxygenates, are available these days at relatively reasonable cost.
Okay, so this will be probably the last question due to time. How is this being accepted by regulators? Can you suggest some paper or report references?
Yes, it's being very well accepted by regulators, and I would suggest that you look at Microbial Insight's website, microbe.com, and if you look at the resources section there, you'll find a lot of different papers that are cited there, as well as ITRC recently put out a publication on the use of molecular biological tools that I think would be very useful to you, including the use of BioTraps coupled with these tools.
Okay, well, thank you, Carrie. That was very informative. For those of you that did not get their questions answered, feel free, like Carrie said, to email him or shoot me an email, and I can get an answer out of Carrie, hopefully, and get back to you. So my information's on your screen, as well as Carrie's. And if you want a copy of the webinar as well, we will have it on our website at eosremediation.com. Also, visit our YouTube channel. Thanks again, Carrie. That was very informative, and we really appreciate you speaking on behalf of us.
All right. Thank you. Bye.
Thanks, everyone.